Good afternoon, everyone, and really excited to be here. This is a very special occasion to be celebrating the Vector Institute, and the things I'll be talking about today really did stem from everything that's been happening in Toronto, of course, for the last 15, 20, almost 30 years now. So big thanks to the organizers. Today I'm going to be talking about BigRNA, which is the world's first foundation model for RNA biology. We all know about RNA. If you think about the COVID vaccine, that was an mRNA vaccine. So RNA basically is what gives life to DNA, if you like. DNA is the same as every one of your cells. RNA is what gives your cells tissue-specific expression, response to disease, and all that kind of stuff. So RNA biology is where it's at, and we're at the forefront of AI for RNA biology. So Deep Genomics, just briefly, our focus, our mission is to decode biology and program life-changing medicines. That's what we do. We have about 100 people, about half of them are computational, half of them are experimental biologists. We've raised $240 million. And I would say that this, Deep Genomics, located here in Toronto, is an example of a big win that's come out of the Toronto ecosystem and the ecosystem like the Vector Institute. So the talk, today I'm going to talk about work that was contributed to by a very large team, of course. I did very little work, but you can see the names of the various people up here. I'll just point out the team lead was Albi Salaj, also Alice Gao, Tammy Lau, and Erla Hogerson did major work on this project, as well as all the other contributors that are listed here. But also I'll mention Tamina Masood, who is our head of target ID, Deep Genomics, and also Shresh Gandhi, who was our head of machine learning at Deep Genomics. So this wouldn't have been possible without this entire team, of course. And many of these people came from the Toronto ecosystem. Some of them came through the Vector Institute. So really, the success of the company is due to this ecosystem. So Deep Genomics today looks very different than it did look 2 years ago because of BigRNA, this foundation model for RNA biology. 2 years ago, we had 40 different machine learning predictors, one for each aspect of RNA biology. And you can imagine these different aspects of RNA biology are accounting for things like transcription or translation and all these other different important processes that are involved in normal and disease biology in every cell of your body. 40 different machine learning predictors. Each one of them had its own training set that had been carefully engineered. They were trained separately, not synergistically. Quite often, to create a training set for one task, we had to subtract off confounding factors, which were in fact confounding factors that the other tasks were meant to solve for, and yet we were training these models separately. As you could imagine, it was a big, complicated network of models feeding into one another. And so a few comments here. First of all, we could not scale that up. It was just too brittle, too complicated to scale up easily. Second of all, each and every one of these 40 data sets required a huge amount of engineering effort to put it together. So just the resources to create those 40 models was tremendous, and again, not scalable. And third, there was no synergy between these different predictions. So that's why BigRNA has been transformational. We now have one model that does it all, realizes synergy between these tasks, and we can scale it up. And really, that's the focus of Deep Genomics in the next year or two, is to scale it up 100-fold in terms of both compute and in terms of proprietary data sets that we're generating and feeding into it. I was at a fireside chat with Jensen Huang, the CEO of NVIDIA, last week, and a few other people that you would know the names of. And I pointed out that I hadn't spoken to Jensen in 7 years. The last time I spoke to him was 2017, and that is why. Really, between 2017 and 2022, Deep Genomics was not ready to scale up. But with this foundation model, we are ready to scale up. So let's get going. This is a scientific meeting, so let's talk about the science. Oh, picture of the team. They're all amazing. Couldn't have done it without them. So just a brief summary to refresh your mind in terms of the central dogma of molecular biology, how it works. So there's DNA. DNA gets transcribed and processed into RNA, which then gets translated and folded into a protein that carries out different functions in your body. And then outside of this, there's cell signaling, there's tissue development, there's physiology, and a lot more. But this is really what's going on inside of your cells. And as I mentioned before, what's critical about RNA is that's where tissue regulation occurs, that's where temporal regulation occurs, time series, and that's where disease regulation occurs. So RNA is really the perfect intervention point for therapeutics and also for just understanding biology so that you could develop drugs to make therapeutics. So the next slide, what I want to talk about is just take a step back from the biology, talk about something we're all familiar with already, which is foundation models. And here's what you get from Wikipedia, at least a couple weeks ago, maybe it's changed. But anyway, this is the definition of a foundation model, a model that's trained on a huge amount of data and then can be applied to a wide range of downstream tasks. And those are the 40 different tasks I was talking about where we realize synergy. And so if you know about ChatGPT for human discourse, AlphaFold for protein folding, well, BigRNA is the world's first foundation model for RNA biology. And just to give you another reference point in terms of foundation models, this chart is showing you on the x-axis time, on the y-axis the number of parameters of the model. So more parameters, more intelligent, maybe. But that's how we think about it. So more parameters is better in certain respects. And first of all, notice on a log scale, note also that if you plot a whole bunch of examples of models over time, they're growing on an exponential scale on that log scale. So it's doubly exponential. But you can see a few different points here that are interesting. Here are some biology results like target scan, splice AI from Illumina, AlphaFold from DeepMind, other techniques like Google Translate. And then there's ChatGPT2, ChatGPT4. So our initial version of BigRNA, which we released last September, is comparable to ChatGPT2. As I said, we're scaling it up, and our current model is around here, and we aim to be up at this scale within about a year. So that's kind of just taking a step back and looking at it with respect to other kinds of models that are out there. All right, so back to the biology. If we look at this process of going from DNA to RNA to protein, here are a couple examples of other foundation models. First of all, we've got AlphaFold, which tells us how to go from amino acid sequence, which is RNA, to folded protein, also ESM fold. And then there are models that don't really capture processes, like going from RNA to DNA or DNA to protein, but they just model the sequence or the biology within a certain slice. So examples there would be single-cell GPT, Bo Wang's group here in Toronto, which just models RNA directly, single-cell RNA measurements. So it doesn't do this mapping or it doesn't look at protein. Atom1, GeneFormer are other examples of that. And then over on the DNA side of things, we have DNA-BERT, Hyena DNA, and nucleotide transformer. Now, BigRNA is the only model that really brings all of this together and predicts RNA biology. So BigRNA, you can take a DNA sequence and predict how the RNA biology will play out in different cells or different diseases. BigRNA also takes into account protein expression, which proteins are expressed, and how they will regulate that process itself. So a major leap forward, really, in the field. Now, I love this slide because it's a bit controversial. Some of you might nod your head, some of you might think it's nonsense. But anyway, some cool things that foundation models can do. Solve tasks without training on any data for that task. So kind of zero-shot learning or something like that. And that's because they've learned something fundamental about what's going on in that area that they were trained in. Achieve higher accuracy than a task-specific AI or machine learning system. So I mentioned those 40 different models. BigRNA works tremendously better on any one task than the models we had trained before on that individual task. Also identify cause and effect relationships. Now, it depends on how you train the model. You have to train a model carefully, but if you do it correctly, then it does identify causal relationships. And all of the models we build at Deep Genomics and BigRNA identifies causal relationships. And what I mean by that is BigRNA can spit out a mutation, and it'll tell you this mutation is going to lead to loss of the protein. You can then go do an experiment in the lab, insert that mutation into a gene in a cell, and you'll see loss of the protein. So it's causal. And then the last one I like a lot, surprise the inventors and users by doing the unexpected. And I think that's sort of what's been wonderful about AI, machine learning, deep learning the last 5 years, is kind of the unexpected outcomes of the models we built. So here's how we trained it. Here's the training data we used to train it. I won't go into the details of how we trained it, but it's trained on over 1 trillion signals, which in the area of biology is truly an astoundingly large amount of data. So that was the key here, is a very large amount of data. 880 genomes, over 3,000 different RNA-seq data sets for different tissues and so on, 51 tissue samples within that data. Also, I mentioned before proteins, so it's trained on protein data sets and microRNA data sets, other kinds of data sets. The figure on the right basically is showing you, I'm kind of unwinding that picture that went from DNA to RNA to protein, showing you some of the steps in between. And so in particular, we go from from the DNA or the genomic sequence up here to the pre-mRNA, which then gets processed into mRNA, which then gets translated into protein. And there's a whole bunch of different processes involved here, as you might guess. There's proteins that bind to the RNA, proteins that bind to the pre-mRNA. We have microRNAs that bind to the RNA that will regulate it. The chromatin in the DNA has structure, so the chromatin can open and allow things to come in and then express a gene, or it closes and prevents it. And so there's a lot going on here with these different processes, and by being trained on this huge amount of data, BigRNA can predict all of that. So everywhere you see a little logo, that's a mechanism or a process that BigRNA can make accurate predictions for. All right, so here's the kind of the block diagram, if you like, of BigRNA. And just at the highest level, to motivate how you would use this system, imagine you could conduct any molecular biology experiment you wanted instantaneously and free of charge. And that's what we're seeking to do with BigRNA. So if you're wondering what would happen with a person if you inserted a mutation, how would it affect their cell biology? Or if you're wondering what would happen if you edited their DNA or edited their RNA, or if you're wondering what would happen if you inserted mRNA, kind of like the COVID vaccines, or you're wondering about a small molecule and the effect that it would have, or an oligonucleotide, the effect that it would have, the idea is that you can just run that through BigRNA and it will make a prediction for what will happen in the cell. And not just a simple prediction, but sort of everything you could want to know about cell biology, molecular biology within that cell. So as you can see in the block diagram, BigRNA takes a sequence as inputs. That could be a DNA sequence or a pre-mRNA sequence, which is translated from DNA. Then BigRNA will analyze that, and it will output tracks that are aligned with the DNA, okay, for over 3,000 different things you might want to look at. And so, for example, up here we have RNA-seq, which is basically gene expression tracks for every cell you might want to look at of these 54 different tissue types. The model can also predict protein binding, so that's shown here in green. So in this particular track, BigRNA is telling you where a particular protein will bind to that RNA sequence or the DNA sequence. Methylation, so when I was talking about modifications of the structure of the chromatin, these are modifications, and there's a variety of different modifications, but RNA methylation is one such modification, and BigRNA can predict where those RNA methylations will occur on that sequence. Now, what's important here, just as you all know, the wonderful thing about AI machine learning is that we seek to be able to generalize. So we train on a certain set of data, but then we can show it a new example, and it can make accurate predictions. And so in the case of BigRNA here, we can train it on half of the human chromosomes, and it can make accurate predictions for the other half, for genes it's never seen before. It can make predictions for a disease mutation, say for spinal muscular atrophy. And even though it's never seen that gene or never seen that mutation. So that's the advantage of having this kind of a foundation model. Now, connecting the dots essentially between these tracks here, these molecular biology tracks, and the more complex outcomes of disease, like pathophysiology, or whether or not a person has spinal muscular atrophy, requires additional information. That's not included as part of BigRNA. But if you think about, people talk about UK Biobank and huge numbers of genomes that have been sequenced. Traditionally, what you do is you look at every mutation in that large amount of data and correlate it with an outcome. Try to correlate it with obesity, with the hope of finding a mutation that's related to obesity. With BigRNA, what we would do is we would take this sequence, all of those genomes, we'd run them through BigRNA, and now we'd be looking at correlations between what actually matters and obesity, which is changes in metabolites, which are transcribed from certain genes, or changes in proteins and things like that, the molecular biology that would drive the disease. So on the next slide, what I wanted to do is kind of give you a glimpse into the breadth of applicability of BigRNA and also the careful validation we've done of BigRNA. Because I think a common understanding out there is people come up with AI and they say it can do amazing things, but does it, right? So, sometimes you'll see these hairballs. A favorite diagram you'll see in molecular biology and cell biology is a diagram with all of the genes on the page in different colors and lines between them. We call those hairballs because you look at that and do you really understand anything about biology or disease or the development of therapeutics? Maybe, maybe not. So what I want to do is just take you through some examples to show you that BigRNA is the real deal. It actually outputs causal interventions that you can go and test in the lab and that do work. We have a lot of results Take a look at the paper that we published in bioRxiv if you want to see the details. I won't be able to go through them all today. But we looked at 12 different drug discovery tasks in detail in that paper and provided validation data. Today I'll just cover a few of them. So the first one is at a very basic level. I mentioned the BigRNA can predict expression for each and every little segment within a gene. And that's what I'm showing you here. And so what we've done here is we've taken a BigRNA, and it has not seen this gene before. We're showing you in gray here, these are the introns and the exons and the UTRs for that gene, so the different parts of the gene. But basically, the gene's roughly 60,000 nucleotides long. And then for each position in there, we use RNA-seq to measure how much of that segment of the gene appears in a particular tissue. I guess it was hypothalamus, okay? And that's shown in pink. So that's a measured data shown in pink. And then what you're seeing in teal is a prediction from BigRNA. And it pretty much, it's surprisingly and shockingly accurate. When I first saw this, I really didn't quite believe it. First of all, it gets two things right. First of all, it tells you which parts of that gene are actually going to get transcribed and processed and stabilized into RNA sequence. But then the second thing is it's getting the overall expression correct as well. In the old days, 5 years ago, we would normalize each gene. So you'd look at the number of reads or the expression of an individual gene, and you'd normalize your predictions and normalize your measured data for that gene. With BigRNA, we don't. The only normalization is the total amount of experiments you've done for the entire genome. We do normalize for that. But it's able to predict the gene expression level and also the structure of what's going on with the gene. Again, this was not the sort of thing you'd believe was doable just 5 years ago. And here's the large-scale data. Of course, people in the room here are into statistics, not just one-off examples. So here's the large-scale data. On the left, what you're seeing is predicted coverage versus observed coverage for a single tissue. So this is brain or hypothalamus. And it's on a log scale, but you can see we get a correlation of 0.74. And again, think about this. It's predicting expression for a gene it's never seen before. And so we can get a correlation value for each tissue. And then on the right, what we're showing you is a histogram where we're looking at bins for Pearson correlation and the number of tissues that have that correlation. And so you can see there's a big cluster up there at around 0.75. And then some of them have lower correlation down here at around 0.5. So some tissues it's not as good at making predictions for. And it's not always perfect. It does sometimes make some errors. A couple kinds of errors that you can see here. First of all, we've got errors where the held-out RNA sequencing coverage predictions, for example, this is shown in the pink, is missing completely. And so here we've got expressed RNA that the model doesn't capture at all. We can also have the other kind of thing happening where we predict a higher level of expression and the observed or measured level of expression is lower. We also see things like intronic regions. So this is in between two exons. On this intronic region, the model predicts there's a bunch of expression going on in that intronic region, whereas the measured data is very low. So those are what we would call errors in terms of the plots you just saw. I'll just briefly point out, though, that quite often errors in prediction in biology is where the good stuff is. And so we've gone and looked at some of these so-called errors and then found that the model is correct and the data was wrong. And what I mean by that, you might be wondering, how can the data be wrong? Well, think about it. An example of this would be, suppose I know the data is on a line, and you got a bunch of data points like this, fit a line through it, and if you know the data came from a line, the line that you fit will be more accurate than the data that you use to infer the line. So that's what's going on here. The BigRNA is actually able to learn biology well enough that it's able to remove measurement errors that are in the data. And then we've gone and validated those kinds of things experimentally and found them to be true. I'm not saying all errors are like that, but some of them. We'll look at another example here just to show that there's tissue-dependent expression. So we're looking at a particular gene here, and we're looking at two tissues, brain and liver. And in this chart here, in the top, you're seeing the BigRNA prediction. And again, so liver is shown in pink, and brain is shown in blue, different from the previous diagram. This is all prediction. And so you can see, for example, that the prediction says that there's a segment here that will be expressed in brain but not expressed in liver. That's what the bump is. And then when we go and do the measurements, look at the data, that also holds. There's a big bump in expression here in brain, but not in the liver. Now, I'll go through just a couple other examples, but I mentioned that RNA is regulated by proteins and other kinds of molecules like microRNAs. And so being able to predict RNA biology crucially relies on our abilities to do that. What I'm showing you here is microRNA binding, one of these important regulators for a gene FBXW7. And in this case here, what you're seeing in orange is predictions of where the microRNA will bind using the state-of-the-art method in the industry called target scan. And so you might imagine applying a threshold or something to just pick up the peaks. And then you're also seeing the predictions from BigRNA at the top there. And you can see that BigRNA has pretty clearly defined bumps where it predicts that the microRNAs will bind compared to target scan. And then what you're seeing down here is validated microRNAs. So these are microRNAs that are known to bind and regulate that protein, that gene. And you can see that the validated microRNAs do overlap with the bumps predicted by BigRNA. And what we can do now is just say, okay, well, that was for one microRNA. Again, let's look at the statistics. So here we looked at a whole bunch of different microRNAs. And for each of those microRNAs, we plotted an ROC curve in terms of our ability to predict the validated microRNA binding sites. And then we measured the area under the ROC curve, the AUC. And so here on the x-axis is this other method, target scan. On the y-axis is BigRNA. So if the methods were performing similarly, they'd be on the dotted line. If they're above the dotted line, BigRNA is performing better. And you can see that in all cases, BigRNA had a substantially higher AUC compared to target scan. We did the same thing for looking at protein binding. And I won't go through examples here, but just wanted to say that we looked at dozens of different proteins and again looked at how where they bound within the gene and then compared it to another technique, this case one called deep ripe. Similar kind of plot. Here it's the average precision, just a different measure was suitable. But again, if they were equal, they'd be on the dotted line. You can see that in all cases, our predictions are substantially more accurate. So now, how would you use this? So we've gone through some stuff that's kind of the statistics in terms of what it's supposed to do when you train the model and things that sort of are akin to the error metric when training the model. I won't go into the details there, but that's sort of what we've looked at so far. But now let's kind of look beyond that and say, okay, I mentioned that this model could be used to do things that are new, to make predictions in a way that would be surprising to the inventors, if you like. So how would you analyze a patient mutation using BigRNA? As I mentioned before, you just put that mutation into the DNA sequence and then run it through BigRNA. So you could look at the sequence with and without the mutation and then just look at what changes. You would look at these tracks and ask what changes. And so, for example, you might see that the patient mutation causes expression to drop to 0 for a particular exon, a particular part of the gene. So that would mean that mutation is causing exon skipping, which could be the cause of the disease. So that would be one way you can use the model. And what's interesting there is we're using BigRNA then to find a cause-effect relationship. If there's this mutation, the exon will be skipped. And we went ahead and validated that experimentally. And so here's just one example where we looked at ATP7B, exon 14, and we looked at a mutation in that gene, which is shown by the little lightning bolt here. And then what I'm plotting is the wild type prediction from BigRNA for expression in that gene. So the wild type has a bump in expression corresponding to the exon. And then also we ran the mutant sequence through BigRNA that's shown in pink, and you can see that the expression level dropped for that exon. And so the prediction here is that there's skipping of that exon due to the mutation. We went ahead and did an experiment where we CRISPRed that mutation into a cell and then did PCR to look at the products, the gene products from that experiment, and that's what's shown on the right. If you're familiar with gels and you're a biologist, then you can see what's going on here. Here's the mutant, here's the wild type, and these two bands correspond to whether or not that exon is present. And you can see that for the mutant, there's a shift towards the exon not being present. If you're not familiar with them, you'll just have to take my word for it. So that's one example, and we've gone through a lot of examples like this to validate that it works generally, but I'm just going to take you through a couple of vignettes. Now, exon skipping is a fairly well-known process. Here's an example that surprised us. And so in this case here, we looked at a mutant, a variant in this gene, NAA10, and we saw that the BigRNA prediction told us two different things. First of all, it told us that the expression dropped. And so again, the variant prediction is shown in pink, the wild type prediction is shown in blue, and you can see that the expression drops. It goes down across the entire transcript. And that corresponds to known knowledge about this gene. It is known that this mutation is disease-causing because you get loss of the protein. You don't get enough protein. What was interesting is that we also saw this happen at the tail end of the gene. The mutation causes a shift over here. You can see a shift from the blue to the pink. What's happening there is the polyadenylation site is shifting. So you might remember poly A, for a gene to have an effect and be translated into protein, a bunch of A's have to be added to the end of the mRNA. It's just one of these things that happens in molecular biology. And where that occurs can be variable. What we found here is that this mutation actually shifts the location of that polyadenylation. That was not something we expected BigRNA to do. It just did it and did it accurately, and we validated that. And I mentioned before, when you're looking at the particular event, like exon skipping or decrease in expression, the BigRNA could also be used to tell you the cause for that. And here's such an example. So the output of BigRNA here, we're looking at two different kinds of output. The first one up here is actually a prediction for a protein. So I mentioned one of the tracks is protein binding. So here what BigRNA is predicting, that in the wild type sequence, the protein has very modest binding, but when there's a mutation in this gene, suddenly the protein likes to bind there, binds really strongly. Concurrently with that, the other track for expression is telling us that when there's this mutation, we see a drop in inclusion of the exon. And so in this case, BigRNA is giving us a bit of a story, a bit of a vignette. It's telling us that the protein binds, and as a result, the exon level drops. It causes exon skipping. And that's shown diagrammatically over here on the right. And so this is an example of what I'm saying about the BigRNA and other foundation models' capability to unlock cause-effect relationships, and in this case, a fairly complicated set of mechanisms, combinatorial mechanisms that lead to the pathology. The other example I'll show you has to do with target discovery. A lot of people are interested in, say, single-cell data or other kinds of data for discovering novel drug targets. And here what we're looking at is the ability of BigRNA to identify pathogenic mutations. So this is a validation experiment first. So we looked at ClinVar, a database of pathogenic and non-pathogenic mutations. And in fact, if you go to see a molecular pathologist and they analyze your mutation or your child's mutation and they classify it, that mutation would be loaded into this database. So it's a database of these pathogenic or non-pathogenic mutations. And then what we did is for each of those mutations, we ran the sequence through BigRNA, and then we used BigRNA to label it as either a mutation that changes molecular biology or a mutation that doesn't, that seems to be benign. So this would only work if the mutation acts through RNA biology, right? Keep that in mind. But that's what we did, and then we'd call that a predicted positive. And then we compared it against the database from ClinVar, which has labels pathogenic or non-pathogenic. And what you're seeing here is the ROC curve. So there's the false positive rate on the x-axis and the true positive rate on the y-axis. The more the curve looks like this, the better. And you can see that the BigRNA predictions are quite good. We've compared it to other methods, but I won't go into that here. Now, how would you use this? So once you've got this capability, we validated it. What's great about this is you have a new mutation, right? Child comes in, or baby in the NICU, new mutation not classified. We can run it through BigRNA and with a certain level of confidence know that BigRNA can give us insight into the mechanism. So we did look at the database, ClinVar, because I mentioned ClinVar has pathogenic and not pathogenic, but it also has ones that are called VUSs, variants of unknown significance. And so in this plot here, these are the VUSs, and these are the ones where the researchers didn't know whether it was pathogenic or not. They couldn't figure it out. And so if we look at these VUSs here, data points right here, and on the y-axis, we're looking at the BigRNA prediction. So the vertical axis is BigRNA thinks that mutation is going to cause something to happen to the molecular biology. Uh-oh, look out. You better check it out, look into it further. And so you can see, for example, here's a mutation that had a very high BigRNA score and was classified as a variant of unknown significance. And so that physician should really look into that mechanism as being, and that variant as being pathological. And then you can see a variety of other mutations that are above the threshold, the dashed line here, which is our threshold for where we would classify something using BigRNA as being potentially problematic or not. It's also interesting that ones that were labeled as benign, we also label some of those as being potentially pathogenic. And so these databases aren't perfect. They contain errors. And so this is a way to recommend that we take a second look at that mutation. Maybe a mistake was made in the lab or whatever, and sort it out. And eQTLs, how many people in the audience are familiar with QTLs and GWAS, eQTLs? Raise your hands. Okay, yeah, there's like 5 people. I'm going to skip this one for that reason. But happy to take a question. Happy to take a question afterward. All right, so I wanted to skip ahead to the other kind of example I wanted to give about the ability to predict something that was out of scope during training, which is the effect of a potential therapeutic. And so in this case, it's an oligonucleotide. And this, what I'm going to tell you here is a bit mind-blowing. What we did to simulate the effect of an oligonucleotide, which, by the way, is a molecule that comes and sticks to the RNA sequence. That's just how it works. It's like one-to-one matching for the RNA sequence. It comes and sticks to the RNA sequence. The way we simulated the effect of that oligo is that we just looked at that window of RNA sequence, and when we fed it into BigRNA, we just set the inputs to a quarter, a quarter, a quarter, a quarter. So we used one hot encoding, right? So 1, 0, 0, 0 would be A, 0, 1, 0, 0 would be C, 0, 0, 1, 0 would be G, and so on. And so all we did is for each one of those nucleotides that binds to the therapeutic, we just set the entries to a quarter. That's it. So BigRNA has never seen oligo data. Never seen oligo data. It's never seen the genes we're going to make predictions of those oligos for. But it's going to work. So now again, just to run you through how you would apply BigRNA, so we'll take that oligo. We find that we just do Control F on the genome to find out where it binds. And once it's mapped up, we just set those inputs to a quarter each. We run it through BigRNA. We see what happens at the outputs. So here, for this particular oligo, it predicts that this exon is going to be skipped. It also predicts we're going to lose a protein binding site. And it also predicts we're going to lose RNA methylation. So that's pretty wild, right? Now we're simulating the effect of an oligo. It's telling us all this stuff about molecular biology. So does it work, right? And this is this example of zero-shot learning. Let's take a look at whether or not it works. So first of all, I'll show you an example, and then I'll show you some statistics. So we looked at ATP7B, and it's the example I showed you before where that mutation causes skipping of the exon. And then what we did is for each position in this region around this exon, we used BigRNA to simulate the effect of a potential oligo there, and we just looked at a whole bunch of oligos across that entire region. And then on the vertical axis, we're predicting the effect of that oligo on exon inclusion. And so that's what's shown on the vertical axis. And so there's kind of 2 peaks here. There's one peak over here, so oligos here are predicted to restore the exon, and oligonucleotides over here are predicted to restore the exon as well. And then we went and tested all of those in the lab, experimentally. And, I'm not going to show you the data for it. Okay, well, you just have to take my word for it. So we went and tested all of those oligos experimentally and found that the actual measured effect of the oligo had a correlation of 0.91 with the predicted effect, 0.91. So incredibly accurate. And the best oligos actually that we found experimentally were located in this region, and there were some that were further down the list over here. So just what you'd expect looking at this plot. And I am showing you one of them over here. So I mentioned we found some over here that were correct and also some over here. So I'm showing you the effect of one of those oligos. So again, the kind of plot you've seen before. What we're showing here is in blue is the wild type. What we're showing here in pink is the prediction from BigRNA due to the mutation. So it's exon skipping. And then what you're seeing in green is the mutated sequence plus the oligonucleotide. And so you can see that expression is restored to the original level. Okay, and here's another example where I'm actually showing you some data. So I gave a talk on BigRNA at the premier meeting on oligonucleotides in October 2023, just a few months ago. And just a few months before that, somebody had published some data where they had targeted a particular gene with a whole bunch of oligonucleotides to develop a therapy for a pediatric patient. So this is data fresh off the press. So we took those oligos, ran them through BigRNA, and made predictions within basically a week or two of submission of the paper. And what you're seeing here is that data that was hot off the press. And so again, similar pictures before. The sequence here shows you the exon and the intronic regions. The lightning bolt is the mutation. And then the blue region here was the best SBO that they found. And they looked at 516 different oligonucleotides. And we then ran all of those oligos through BigRNA, and the best oligo they found was ranked among the top 7 in terms of predictions from BigRNA. And the scatter plot is shown on the right. So here's the BigRNA prediction, and here's the efficiency measured in the wet lab experiment. And I mentioned before I'd tell you some statistics about our ability to do this. And so we've looked at 15 different exons and 12 different genes to ask whether we could predict these oligonucleotides. And that's what you're seeing here in this plot. On the x-axis, we're just looking at correlation between the predicted and the measured exon inclusion levels for all of these 15 different exons. And then if we look at another technique that's out there, Splice AI from Illumina, you can see the predictions that it would have made, and you can see the BigRNA generally performs substantially better. The last example I'll give is one that has to do with restoration of protein. So exon skipping is one particular kind of therapeutic that's advantageous. There are a lot of diseases that have to do with haploinsufficiencies, where you've lost one copy of your 2 genes. You know, you got 2 genes, one from mom, one from dad. One of the genes didn't show up. You've only got 1 copy, and so you've got half the amount of protein you need. So the question is, can you take that protein that's still working and boost it? So introduce a therapeutic that will double or triple the amount of protein being produced from that 1 copy. So that's how we would deal with what's called a haploinsufficiency. And so here, in terms of the BigRNA prediction, what we're asking for is, would the oligonucleotide cause a general shift upward in the expression level? So in this case, it's kind of roughly doubling the amount of mRNA being generated by that gene. So if we can find a therapeutic, whether it's a DNA edit, an RNA edit, or some other therapeutic that can do that, we could address that haploinsufficiency. And so the last example here that I'll just take you through is the experimental data for doing just that. And what we've got, it's a bit of a different plot than before, but we've got the BigRNA prediction, inhibitory score, so high is good, low means BigRNA thinks it won't do anything for that oligo. And then what we're looking at is the active SBOs, and those are the SBOs that we've carefully validated with dose response curves and so on that actually do have the desired effect on that gene in vitro. And what you're looking at over here called other SBOs is all the other extra SBOs that were that were designed without using BigRNA. We wanted to have a control set in there to make sure that we weren't missing things that BigRNA missed. And so you can see that the control set's way down here. The active SBOs are way up there in terms of the predicted scores. And then we see that for 3 other genes we looked at. So that one's PON1. We also saw the same thing for ATP7B, for PRRT2, and for SERPING1. Okay, so that's it. So hopefully I kind of, first of all, introduced you to the idea of the foundation model for RNA biology. I convinced you that this BigRNA kind of really can enable you to conduct kind of any experiment you might want in the computer. And in terms of where we're headed, we've covered sort of these we've checked off these goals in terms of what we're trying to do: rapid exploration of RNA biology scale, in vitro validation. We've got that. Support for multiple therapeutic modalities. 2024 is about a few different things. One of them is in vivo validation, so doing mouse and NHP experiments for these therapeutic modalities. And then where we're headed in the future, I mentioned this before, but really 100-fold scale up. So that has to do with proprietary data, increasing model capacity, expanding the function of BigRNA, and increasing its accuracy. A big part of that, of course, is just hardware, having 100 times more hardware. Also an important one is improving mRNA resolution and cell modeling. So the public version of BigRNA can only make predictions that are 128 base pairs when it comes to gene expression. That's why those plots kind of look chunky. But we're building a 1 base pair model. It can make very, very accurate predictions. And then also look at things like single cell data and cell state. And then finally, validation data for DNA and RNA editing and also mRNA. All right, thank you very much.
All right, we have time for 1 or 2 questions. Why don't we start with one online question first?
Hi. One question we have is, what could BigRNA have an impact? What impact would BigRNA have on personalized medicine?
Yeah, great. So what impact would it have on personalized medicine? One of the applications that Deep Genomics has so far not been able to focus on is very personalized medicine, which is N equals 1. I mentioned one of the examples there where I said the physician had developed a therapeutic for a pediatric patient. N equals 1 medicines are where you literally have a baby in the NICU. The baby will not make it past a year if the mutation is not treated. So it's not really a good business opportunity. It costs about $1 million to fully develop one of these oligonucleotides into a therapeutic. And yet, of course, BigRNA is the perfect technology for that. Like, it's just perfectly suited to that. And so that's why one of the reasons we made BigRNA public is so that other people could use it for those kinds of applications. We don't know how to make that into a successful business model ourselves. However, there are a lot of patient physician scientists out there, families who are closely involved in a disease for their child or their loved one, who would have the capability of using something like BigRNA to understand the mutation and then also develop a therapeutic. All right.
Maybe.
Thank you.
One question on the floor.
Hey, Brendan, really cool stuff. So for the next successor to BigRNA, what is the kind of training data that you wish you had access to the most? What would be most helpful?
Yeah, great question. So there's two kinds of data, really. One of them is just more of the related data, so DNA sequences and protein, and so we'll use more of that existing data. And then the other one is just, when I showed you the map of all the different RNA biology processes, proprietary data, or data, I should say, that just enables us to train on those processes more accurately. MicroRNA binding is surprising, actually, how there's a dearth of good data for microRNA binding. And so we've generated some data, but external efforts to generate data would be valuable. But that's the way I would think about it right now is, you look at that map of RNA biology, just what kinds of additional data sets can be generated for those. And then another one, I guess, is single-cell sequencing. And so one perspective is data for different mechanisms. Another one is data that's more, that has higher resolution for what you're measuring in biology. So single-cell sequencing data is data that we're looking at. It's unclear, as I'm sure you know, it's unclear how you kind of make sense of single-cell sequencing data just in terms of the relationships between cells. It's kind of a chicken and egg problem. Many cells come from the same tissue, and yet the whole reason you're using single cells is because you don't want to aggregate cells into tissues. And so there's not really a great solution for that yet, but models like single-cell GPT or other related models are helping in that area. But I would say single-cell sequencing is another example. And then more complicated models like organoid models, which capture more of biology than just two-dimensional models where you put the cells in plates and then measure the results. All right.